Hello everyone. Today we're going to talk about different tests that can be used to identify and classify microorganisms. Let's talk about the taxonomy hierarchy that we use to classify organisms out there. So all organisms out there, we go ahead and we classify them under a domain. And we've got three domains. We've got eukarya, archaea, and bacteria. Viruses are the only ones that do not fall into any of those three domains. So viruses are a group on their own. But anything other than viruses, you go ahead and you classify them under a domain, and then you figure out which kingdom they fall under, and then phylum, class, order, family, and then genus and species. And usually when we read the scientific name of an organism, we just include the genus and species in the name. So for example, Staphylococcus aureus. That would definitely be a domain bacteria. So Staphylococcus aureus is a bacteria. And then Staphylococcus would be the genus, and then the species would be aureus, or in this case, Escherichia coli. So Escherichia would be the genus, and then coli would be the species. Now, under the domain eukarya, if we talk about eukarya first, there are multiple kingdoms. So you have kingdom fungi, planta, animalia, protista. Protista, you have those protozoans that, for example, like amoeba and euglena, all of these would be considered eukaryotic organisms. And of course, each organism is going to fall under a kingdom and a phylum, etc. Now, of course, organisms that, let's say, fall under the same kingdom, they will have a bit more similarities. If they fall under the same phylum, then they are even a bit more related, and they have even more characteristics common. So as you go further down, if organisms fall, for example, under the same family or under the same genus, then the more common characteristics they're going to share together. Under the domain archaea, you would have organisms. Those organisms, they are microorganisms. You'd have to view them under the microscope. We have different types of archaea, microorganisms out there. A few examples, you have methanogens, and those are microorganisms that produce methane gas. You also have extreme halophiles. So those are microorganisms that love to live in regions that are extremely salty. You have hyperthermophiles, and those are microorganisms that like to live in temperatures that are really high. We usually don't discuss archaea that much because till now we haven't figured out if there are any archaea that are actually pathogenic to humans. And so again, we don't really discuss them too much. Bacteria, on the other hand, because there are lots of bacteria that are actually extremely helpful to humans, and some bacteria, they are extremely pathogenic and virulent. We give them more of our time and research to figure out, again, how we can benefit from those beneficial bacteria, and then how we can get rid of those pathogenic ones. And again, with each bacteria, you would classify it under the kingdom, the phylum, etc. And then, as I said, the genus and species are usually what we would use to, to have the scientific name of that microorganism. The genus and species is what's going to be written down. So let's go ahead and move on. Now, to classify those microorganisms, we classify them based on a lot of things. One thing is based on their morphology. So how do they look like? Morphology and arrangement. So, for example, if we are going to discuss bacteria, we go ahead and we create a bacterial smear, we look at it under the microscope, and then we take a look at its morphology. Is this bacteria spherical? Then it's coccus. If it's rod, it's bacillus. If it's spirally, it's spirillum. And then you go ahead and you also take a look at its arrangement. For example, are those coccus bacteria that you're looking at, are they in a chain? Then we would refer to it as a streptococci bacteria. If they are clustered together, you would say staphylococci. So again, based on their morphology and arrangement, that's the first step in identifying which type of bacteria you have on that smear. And then we go ahead and we do those differential staining, like a gram stain and acid-fast stain. So the gram stain is going to tell us whether our bacteria has a thick peptidoglycan cell wall or whether it has a thin one. If we do the acid-fast stain, it's going to tell us whether our bacteria has a waxy lipid layer on its surface or it's not going to have it. And again, we're going to figure that out when we do the stain and we get different colors. Then we are able to tell whether our bacteria is gram-positive, whether it's gram-negative, acid-fast bacteria, non-acid-fast, etc. There are some bacteria actually out there, not a whole lot, but they don't have a cell wall. So if they don't have a cell wall, then the gram stain and the acid-fast stain are going to be not useful to be able to differentiate between those bacteria that don't have a cell wall. We also do a bunch of biochemical tests, and those biochemical tests, again, would help us identify the bacteria because it would tell you if those bacteria have some special enzymes, and based again on the special enzymes that they have, we can go ahead and try to figure out and differentiate between the different bacteria. To identify, let's say, different bacteria, we go ahead and we create what's known as a dichotomous key. A dichotomous key is basically a bunch of questions you would ask, and the answers are always either yes or no. And based on the answer, you go ahead and you follow it up with another question, and you keep asking questions until you figure out, okay, what is the bacteria you are looking at. So let's say that we have a plate and we cultured some bacteria on that plate, and let's say the plate is containing the sugar lactose. Now, not all bacteria are able to actually break down and ferment lactose. Some bacteria are capable and some bacteria are not able to break it down. And so that lactose agar plate is actually a differential plate because it is going to help you differentiate the bacteria based on can they ferment the lactose or they can't ferment the lactose. And how can we know that? How can we tell if the bacteria was able to ferment the lactose or not? The plate color is going to change. So the media color is going to change. And so you can start asking a question, okay, can those bacteria ferment lactose? Is it a yes or a no? If it's yes, then you can move on to like another question. If it's no, again, you usually kind of ask the same questions and you keep asking the questions, okay, so can they ferment lactose, for example, can they ferment glucose, are they gram positive, are they gram negative, under the microscope, are they cocci, or are they bacilli? And again, you keep answering the questions yes, no, until you go ahead and you figure out the bacteria that you are looking at. So right here, you can ask like another question, okay, can they ferment sorbitol? Sorbitol is a sugar alcohol. And again, based on your yes or your no, you can figure out the bacteria. It takes more than one or two questions, okay? So the dichotomous key, it's going to be a bunch of questions before you go ahead and you figure out what type of bacteria. And of course, you're asking those questions and you're figuring out the answer based on the results that you're getting. So you're not making any hypotheses or any guesses or anything. It's based on the results that you're getting from your culture plates, from those biochemical tests. You are able to figure out whether the answer is yes or no. One of the biochemical tests that we do, it's known as a rapid identification test, and it basically tests several biochemical tests all at once. So that's why we call it rapid, because you get so many results all at once. And so what we do is we get a test tube, and this test tube has media, and you can see that the media within that test tube is divided into several parts. Part of this media, for example, has the sugar glucose. This part of the media has lactose. This one has sorbitol, etc. And then what we do is we get an inoculating group, and you take some of your bacteria that's, let's say, been growing on a plate. So you go ahead and you take some of that bacteria, and you go ahead and you swab it basically, and you inoculate it onto that test tube that has the different media, and you leave it to incubate, and then you go ahead and you take a look at the results. You might start seeing color changes. So based on the color changes, you would figure out, okay, this bacteria was able to ferment glucose. This bacteria was able to ferment lactose. This bacteria was able to produce, for example, hydrogen sulfide. And so based on the results that you get, we usually go ahead and we give it a number. So there is usually like a list of numbers, and they would tell you, it's kind of like you would use a key, and they would tell you, okay, if it does ferment glucose, or if it does produce hydrogen sulfide, it's you know, it's a number 2. And so you would keep, like, you know, looking at the results and choosing the numbers accordingly until you get a code. So here's the code right here. And then you take that code and you take a look at a database that, a computerized database that they have, and you compare. So you go ahead and you go like, okay, this bacteria is a 62353, and then you compare it with the database on the computer, and they would tell you, okay, this is the bacteria that you have. So that's a test that's usually done. Serology is basically the study of serum. So what is serum? First of all, serum is the liquid part of the blood that contains antibodies, antigens, doesn't contain cells, and it does not contain proteins. If you are going to think of the liquid portion of your blood and you're thinking that it has antibodies, antigens, plus proteins, like proteins that can clot your blood, etc., that would be the plasma. So that's the biggest difference between a plasma and a serum. So we usually use a patient's serum that contains antigens and antibodies to figure out which type of bacterial infection do they have, because bacteria have antigens. So microorganisms, we refer to them as antigenic microorganisms. And so when a microorganism is introduced in your body, your immune system is going to try to fight it. And one way it goes ahead and it fights it is your body is going to start producing specific antibodies against those antigens. And those antibodies are going to help you basically fight off those microorganisms. And so one way we go ahead and we figure out what type of bacterial infection we are infected by, we go ahead and we, let's say, place some bacteria on a slide. Okay, and let's say we know what type of bacteria that is. So we go ahead and we spread some bacteria on the slide, and then we go ahead and we take the anti-serum, which is basically the serum of a patient that has some antibodies, and we go ahead and add it to it, and we see what happens. So let's go ahead and actually take a look at an example. So here's an example right here. Here's the slide, okay? And on the slide, they've smeared a bacteria, okay? So they went ahead and they smeared the bacteria. Now, you could start off knowing what bacteria you're smearing, okay? So I am smearing a specific bacteria that I know. And then you go ahead and you take the patient's serum, and you're wondering whether the patient has this bacterial infection. And for you to figure it out, when you're extracting the patient's serum, if they do have the infection, if they do have that specific bacterial infection, then their body has already produced antibodies against it to help them fight it off. And so if you go ahead and you drop some of the patient's serum on the slide, if the patient has the antibodies, then those antibodies are going to quickly react with the bacteria already on the slide, and what you're going to see is a positive test. The bacteria is going to start to agglutinate. It's going to start to clump up because there's a reaction taking place. If the patient doesn't have the antibodies, meaning they don't have this infection, then nothing is going to happen on the slides. And so this test is either done by placing first, let's say, a swab of a patient's, let's say, throat, which might have bacteria, and then you go ahead and you smear it on the slide, and then you go ahead and you add a known antibody. So you can either do it like, either way, either put the bacteria, a known bacteria on it, and then put the patient's serum and see whether the patient has antibodies against it, or you can put the smear of the bacteria you got from the patient first, and you have no idea what bacteria they have, and then you go ahead and add to it known antibodies. And in both cases, if you have agglutination, that would tell you that this patient has this bacterial infection. Another test that's done is known as an ELISA test. ELISA stands for Enzyme-linked immunosorbent assay. We usually just say ELISA for short, and you can see right here how an ELISA is done. So you have a microplate right here, and then you start micropipetting a sample. And we're going to talk about what samples are being micropipetted right here. And here is a zoomed in image on the microplate ELISA. It has so many small wells, as you can see, and you go ahead and you pipette your sample in those wells, and then we go ahead and we use a computer scanner to scan the results for you, and it would tell you the results. So what are we really micropipetting into these little wells? It's the following. First of all, there are two types of ELISA tests that we could do. One is known as a positive direct ELISA, and one is known as a positive indirect ELISA. The positive direct ELISA is used to detect if you have a certain antigen in your serum. So again, an antigen in this case, meaning, for example, if you have a specific bacterial infection. Again, foreign microorganisms, we refer to them as foreign antigens. So again, here, when you see the word detect antigens, you can think of basically a bacterial infection. So again, the positive direct ELISA is going to confirm whether you have that bacterial infection. Now, how is it going to confirm if you have that infection or not? Here is the well. So here's a zoomed in image of the ELISA well. So what we put in the well is antibodies. Known antibodies. So exactly, like, we know which antibody are we adding. Again, antibodies are different. So you have a different antibody for each different antigen. It's not like an antibody can bind to whatever antigen. So you know for sure that this is a known antibody, let's say against bacteria A, okay? And you want to figure out whether the patient is infected with bacteria A. So what you do is you go ahead and you take a sample from the patient, and you go ahead and you add the patient's sample. Now, if the patient does have bacteria A, it is going to bind to that antibody because this antibody only binds to bacteria A. And now, let's say this patient has more bacteria B, bacteria C, it's not going to bind because this antibody is only specific in binding to a specific antigen. So now, after the antigen binds to that specific antibody, what we go ahead and we do is we go ahead and we pipette another antibody. And this antibody is a bit special because this antibody is again specific to binding to that antigen. But what's special about it is we went ahead and we added enzymes. So we've attached enzymes to those antibodies. Then what? We know that enzymes have active sites. Right here, you see those active sites. What binds to active sites are substrates. So we go ahead and if you see those squares right here, we add the substrate. The substrate is going to bind to the enzyme linked to the secondary antibody that you've added. Once the substrate binds to this enzyme, a reaction is going to take place, and the well is going to change color. So the solution in the well is going to start changing color. So once it changes color, then we know the patient has this specific bacterial infection. Over here, when we added the patient's sample and the bacteria was able to bind to the antibody, notice no change took place. And so just by looking at the well, you're not going to be able to tell the patient has this bacterial infection because no change is taking place. The only time we're going to figure out really that, okay, a change has taken place is after we add that secondary antibody linked with the enzyme, and then we add the substrate. Once the substrate binds and reacts with the enzyme, the change in the color within the well is going to take place. And if that happens, that's a confirmation that the patient has this bacterial infection. The other way of detecting whether the patient has a certain bacterial infection is instead of adding a known antibody first, we go ahead and we add the known antigen. So again, we add bacteria A already into the well. And we want to figure out, again, whether the patient has bacteria A. So what do? We take a sample of the patient's serum. And as I said, the serum is where you will have your antibodies, okay? And again, we don't know whether the patient has bacteria A. If they do, if they are infected with bacteria A, then their serum is going to contain the antibodies. So if you take a sample of the patient's serum and you go ahead and you add it, if they do have the infection, as I said, they will have the antibodies. So notice how if they do have those antibodies, it is going to bind to the bacteria, which is a confirmation they actually have the bacterial infection. But again, we can't really detect it. If we look at the ELISA well, we can't really detect it yet because no color change has taken place. And so what we do is we go ahead and we add the secondary antibody, which is capable of binding to the patient's antibody. And that secondary antibody, as I said, it's a bit special because it has enzymes linked to it. So those enzymes, one more time, they have those active sites. We go ahead and we add the substrates. If the substrate binds to that secondary antibody, which is bound to the primary antibody that was already added from the patient's serum, a color change is going to take place. And again, that will confirm that the patient has that bacterial infection. So as you can see, there are two methods for the ELISA to figure out, if the patient has that specific bacterial infection. Let's say the patient doesn't have the bacterial infection. So if the patient doesn't have the bacterial infection, for example, if we look at this method, then when you add the patient's serum, no antibodies will bind to the bacteria. So it's going to be kind of something like this. No antibodies are bound. Why? Because this patient doesn't have that bacterial infection, and so therefore they never made those antibodies. And so if you add the secondary antibody, So if you add the secondary antibody, it's not going to bind to anything because that secondary antibody only binds to the antibody. And so it's not going to bind to anything. And so therefore, no color change is going to take place. And that would confirm that the patient doesn't have the antibody. This is how we can confirm whether the patient has indeed that bacterial infection or whether they do not. Another method used is known as the Western Blot. This method is used to detect whether an individual has a specific antibody. And if they do have that specific antibody, that would be a confirmation that they were infected by that specific bacteria. So let's say that we're wondering whether a patient was infected, let's say, by bacteria A. So we go ahead and we actually get bacteria A and we lyse it. We break it down. So we get those bacteria, we break them down, and we extract the proteins out of them. Those proteins are basically, we can refer to them as the antigens. We take those proteins and we run them down a gel. So here's the gel, and you run down the proteins. They're going to run down the gel. And then what we do is we want to go ahead and actually transfer those proteins, those protein bands that you're seeing right here on this gel. We want to transfer it over to a membrane. We usually use a nitrocellulose membrane because it has high affinity to proteins. To do that process, we go ahead and we kind of compact the gel by putting on top of it a bunch of filter membranes and sponges, and then we put it in a cassette and it kind of squeezes it, and then we turn on the electricity, and the electricity is going to help transfer over those protein bands from the gel onto the membrane. After successfully transferring over the protein bands on the membrane, what we do is we go ahead and we flood this membrane with the patient's serum. Now, if the patient is indeed infected with this specific bacteria, the patient's serum is going to contain the antibodies. So if the patient's serum contains the antibodies, when you flood that membrane with the patient's serum, if they do have those antibodies, the antibodies are going to bind to those protein antigens that you have on the membrane. But that's not going to cause a color change. So for us to detect really the patient does have those antibodies, what we do, we go ahead and after first flooding it with the patient's serum, we go ahead and we wash it off and we flood it with a solution that has a secondary antibody. And that antibody binds to the patient's antibody. And that secondary antibody is special because it has the enzymes linked to it. Then we go ahead and we flood the membrane with the substrates. Once the substrate binds to the enzyme on the secondary body, a color change is going to take place, and those bands are going to become visible. And they are going to be visible, and for example, they're going to be colored red, for example, and once they are visible, then you know the patient indeed had the antibodies in their serum, and that's a confirmation that the patient did have this infection and they did have this bacterial antigen inside of them. Another test we do is known as phage typing. Phage is short for bacteriophages. What are bacteriophages? Bacteriophages are viruses, natural viruses that have the capability of killing certain bacteria. Again, not all bacteriophages can kill any bacteria out there. They are pretty specific in terms of which bacteria they can kill. Bacteriophage typing the way it's done is we go ahead and we get a plate. And we culture a bacteria on that plate right here. We create a lawn of bacteria. Then we go ahead and we start adding bacteriophages. Now you can add one bacteriophage, two bacteriophages. You can add so many different types of bacteriophages all at once on the plate. Of course, you will have to note where did you add the bacteriophage 1, where did you add bacteriophage 2, etc. And then you leave it to incubate, and then you go ahead and you take a look at your plate. If nothing happens, notice how here nothing happened to your bacteria lawn. That means that none of the bacteriophages that you've added right here were capable of killing the bacteria. That means that the bacteria that are growing right here, they are all resistant to those bacteriophages that you've added. Notice how here you have these plaques. That tells you that actually the bacteria in those regions right here, they are getting killed by that specific bacteriophage. And the stronger looking, like the darker kind of clear area that you're getting on your plate. So it's going to look something like this, like circles, okay? This would tell you, see like how this one is like way darker than that one. So this tells you that this bacteria here, like that bacteriophage is capable of doing more damage to that bacteria versus this bacteriophage right here. Since you don't have, again, as much reaction on this part right there. Bacteriophages are actually getting now a whole lot of attention because there are a lot of bacteria that are now becoming resistant to a lot of antibiotics that we are using against them. There are bacteria that are actually resistant to every single antibiotic we have out there. And so what we could do then to kill those resistant bacteria that are resistant to the antibiotics is try to kill them using those bacteriophages. Another test we do is the fluorescent-antibody technique. Again, if we want to figure out whether the patient has the specific antibodies against that bacterial infection, or whether we want to figure out whether the patient has that bacterial antigen. One way of doing it is we go ahead and let's say we get a sample from the patient's throat, and we are suspecting that the patient has streptococci bacteria. We go ahead and we add to the patient's sample, we go ahead and we add specific antibodies that are capable of binding to those streptococci bacteria. But what's special about them is they are fluorescent. So we've added like a fluorescent dye to those antibodies. Now, those fluorescent dyes will go ahead and they will bind to the bacteria, and the bacteria is going to fluoresce under the microscope. So if the bacteria fluoresces under the microscope, then indeed the patient does have the streptococci bacteria infection. Another way of doing it is we go ahead and we take, like, again, a sample, a bacterial sample, and then we add the patient's serum to it. So the patient's serum, if they do have this specific infection, the patient is going to have the antibodies. Now, the antibodies we produce are definitely not fluorescence antibodies. And so if the antibodies indeed bind to those bacteria, we can't really detect that because we can't see it. And so what we do is we add a secondary antibody that are fluorescent, and those secondary antibodies, they are going to bind to the patient's antibody. If they do bind to the patient's antibody, when you look at it under the microscope, you are going to see fluorescence, and you are going to see those bacteria fluorescing. If, again, the patient doesn't have those antibodies because they were never infected by this bacteria right here, then those fluorescent antibodies, they're not going to be able to bind to, they're not going to be able to bind to anything, because we usually if you're wondering, like, why after adding those fluorescent dye, the whole slide is not going to fluoresce since I've already added them. The thing is, after each step we wash. So after each single step, we go ahead and we wash our smear. Same thing with the ELISA method, same thing with the Western Blot method. After every single step, we go ahead and we wash the wells, we wash the membrane, we wash the slides. And so when you wash them, if those antibodies were not bound to anything, they're going to be washed off. And so therefore, you're not going to see your bacteria fluorescing under the microscope because, again, they were washed off because they were not able to bind to anything. And that's how, again, we can use the fluorescence antibody technique to confirm whether the patient has that bacterial infection. We have a test known as the DNA fingerprinting, and the DNA fingerprinting is basically a way for us to figure out whether organisms are related by trying to figure out whether they have genetic similarities. So one way we do it is we go ahead and we extract DNA out of 2 organisms, and then we break down the DNA using restriction enzymes. You go ahead and you break it down with those enzymes, and you run the DNA down the gel, and then you compare. So notice how those 2 samples right here of DNA, they are basically identical. That would tell you that this organism right here is identical to that organism right there. Let's say they are not completely identical. So if you compare, let's say, this organism's DNA with this organism's DNA, now you can totally tell they're not the same. So they're not the same organism. But if they have so many similarities amongst them, that could tell you that they are probably related. Because again, their DNA is very, very similar. So the more similarity they have, then the more related they are. If they are exactly identical, then, well, you're looking at the same organism. There is a method known as the nucleic acid hybridization. So with the nucleic acid hybridization, again, we use it to figure out whether organisms are related and how much similarity do they have between them. So let's say you have organism A and organism B, they have a double-stranded DNA. You go ahead and you go through a process to basically break down or detach, really, the 2 DNA strands. After you detach the DNA strands, you go ahead and you try to combine them. So you basically mix the DNA strands of organism A with some of the DNA that has been detached from organism B. Now, if you find complete hybridization, like the complementary DNA strands were actually able to bind to one another completely, that would tell you that those organisms, again, they are identical. But if you find partial hybridization, well, those organisms are related. If no hybridization is detected, then those organisms are not related at all. You might wonder, well, okay, how am I going to figure out whether I have complete, partial, or no hybridization? We usually add either fluorescence or we add certain compounds that would be detected when you use an X-ray machine. So let's go ahead and kind of take a look at an example. So let's say you have this bacteria right here, and this bacteria has plasmids, which is basically DNA. And you go ahead and let's say you extract a piece of the DNA from the plasmid, and let's say you want to make more of it, so you want to clone it. You can actually insert it into another bacteria, and as the bacteria is growing, it's actually going to clone that piece of DNA for you. And then what we usually do is we extract the cloned DNA fragments that we have, and we go ahead and we mark them with fluorescent dye. So now those DNA probes are fluorescing. Let's say that you have an unknown bacteria growing on a filter. You go ahead and you extract their DNA. So here's their DNA, and you go ahead and you break it down. So again, you basically, well, I shouldn't say break down their DNA, I should say you're separating their DNA strands. So not breaking down their DNA strands, you are separating their DNA strands. So here we go. Now, notice how you might actually have more than one bacteria growing. Again, if you culture some bacteria on a plate and then you kind of look at it, you might actually see different bacteria growing on your plate, which means you have different DNA. Therefore, when you go ahead and you split the DNA up, you might have different strands of DNA. Now, let's say I want to figure out whether right here I have salmonella. How can I tell whether I have salmonella or not? One way of doing it is we go ahead and we take the salmonella, now fluorescent, right, DNA strands, and we go ahead and we add it to the single strands of DNA that we have. If we do have salmonella, then this fluorescent probe is going to hybridize. It is going to bind, and you're going to have the hybrid now between that original DNA that you got from your plate and that fluorescent probe. When you look at it, it is going to fluoresce, and that will tell you that on this plate you do have salmonella. The bacteria or the DNA that's basically not going to fluoresce is going to tell you, well, okay, this DNA is coming from another bacteria. It's not coming from the salmonella bacteria, and therefore the fluorescent probe was not able to bind to it, and therefore it's not going to fluoresce. There is another blot that we do known as the southern blotting. And southern blotting is, again, we are trying to figure out unknown microorganisms using fluorescent DNA probes. Or sometimes those DNA probes are not fluorescing, but you can detect them using an X-ray. And so what we do is, again, we use a gel electrophoresis. We allow the DNA of that microorganism to run down the gel, and you start getting DNA bands. We then transfer the DNA bands that we get on the gel onto a filter. So right here, the DNA bands would be on the gel, and we go ahead and we transfer them over to a membrane. Then what we want to do is we want to figure out whether those DNA bands that we get belongs to, let's say, again, the salmonella bacteria. So how can we figure it out? We go ahead and we take, see those like red probes right here? So you go ahead and you flush that membrane with the salmonella DNA probes that you have. If you do have the salmonella DNA, then those probes are going to bind to it. So they are going to hybridize to the complementary strands that you already have on your DNA. And then we can actually take this membrane and you would put it and use an X-ray machine. So you go ahead and you use an X-ray machine and you get your film, your X-ray film, and you look at it. If your X-ray film shows nothing, then that means that no hybridization took place, and that means that you don't have the salmonella DNA on your filter. But if looking at the X-ray film you actually see bands, that would tell you that indeed you do have the salmonella DNA on that membrane, because the bands are going to become visible. Another way of kind of detecting that DNA hybridization is we do it using a DNA chip. So the DNA chip, again, we add the DNA probes that are usually fluorescent DNA probes, and we add it to a sample of DNA that we have on that chip. And again, if we do detect any fluorescence, then that means that we do have this bacteria on the chip. So it kind of looks something like this. Here is the chip, and you have little wells where you've added samples of, again, split DNA strands. You add them within these wells. And then on those wells, what you add is the fluorescent probes. If the fluorescent DNA probes were able to combine with the DNA samples that you already have on the chip, then that would tell you that you do have this DNA added. Those DNA chips can actually be used to figure out specific genes that are carried. So if we have a DNA probe for, let's say, an antibiotic resistant gene, and it's basically fluorescing red versus another antibiotic resistant gene, it's going to fluoresce yellow. You go ahead and you look at your chip and you figure out the colors that you get. So if it fluoresces green versus if it fluoresces yellow versus red, that would tell you, again, what could that bacteria, like which type of genes it could actually be carrying, which type of, for example, antibiotic resistant genes is it carrying, based on the fluorescence colors that you get.